Hi, everyone. In this video, we will be discussing topics surrounding the ideas of sexual reproduction and meiosis. So sexual reproduction itself involves the production of gametes and the fusion of those gametes to kind of produce a genetically unique offspring. So in this case, with sexual reproduction, it's two parents. Could be like an egg and a sperm in this case. And then by mixing the chromosomes of two individuals, the genetic diversity itself gets increased, and then this process itself is different from mitosis in that one cell basically produces two genetically identical offspring cells. Now, meiosis itself is the division in which cells become gametes. So it goes in two rounds of division, which it ends up with half the chromosomes that you initially have. So, for example, in humans, we have a total of 46 chromosomes, but it ends up coming down to 23 at the end of meiosis 2. And usually it's accompanied by cytokinesis. That way it has 4 separate cells being formed. And during sexual reproduction, a sperm and an egg kind of unites to form a new individual through the process of fertilization. So in this case, where when the homologous chromosomes come together, they have the same genes but different versions. One is probably from the maternal side and one from the paternal side, or like one from mom, one from dad, and then these have the same genes, same location, in the same order on each chromosome, but it could actually have different alleles or versions of that specific gene. So for example, gene for eye color, it could be that one is coded for blue, one is coded for brown, so on. Now, overall, meiosis, like we talked about, it has two sets of divisions, meiosis 1 and 2. And it results in half the amount of chromosomes that you start off with. And the major thing about meiosis is that the chromosome replication only occurs before meiosis 1, and then it will not do that again. So it's basically like 2 rounds of mitosis, except during the second round, there is no duplication of the chromosomes themselves. So the phases of meiosis 1, they include early prophase 1. In this step where it basically goes into synapses, which means that they are coming together and trying to cross over, and basically comes together aligned with the homologous chromosomes, forming a tetrad itself. So this is a tetrad. And then in late prophase 1, what happens is they're still together, but now what happens is the non-sister chromatids of the tetrad itself exchange DNA at the crossing over point. So the 2 homologous chromosomes come together, and then they basically exchange or give each other a part of their DNA to kind of make a new combo. So basically with recombination that typically occurs with non-sister chromatids. Since you can only pass one chromosome to the offspring itself, recombination in meiosis is a way to basically get some genetic information from each chromosome to the other in the pair. That way, no matter which one ends up in the gamete, there will be some uh, parts of the DNA going in from both chromosomes, and that ends up eventually getting inherited throughout. Now, the next phase itself is called metaphase 1. It's basically similar to what we talked about in mitosis, where they align up at the metaphase plate in the center of the cell. And then it goes into anaphase 1, where it pulls apart the homologous chromosomes like this. And then it goes into telophase, where the chromosomes arrive at the opposite ends of this cell, and then goes into cytokinesis, which ends up with two daughter cells that are haploid, which means there are 3 chromosomes instead of 6. Now it goes into what is called meiosis 2. So the cells that are entering meiosis 2 are basically haploid, so half the amount of DNA, and it is not identical. Each cell has a chromosome, basically a homologous pair, which can be from mom or dad in any random order. Now, with meiosis 2, it goes directly into prophase. There is no DNA replication happening. So it goes into that, and the spindle apparatus gets formed like this. It attaches to the centromeres of each sister chromatid, and what happens is in metaphase 2, they end up aligning again at the metaphase plate. The chromosomes themselves end up being in the middle itself. And then it goes into where the sister chromatids are now being separated by anaphase 2. And then it goes into telophase, where it arrives at the poles and separates out into 4 unique little cells after it goes into telophase and goes through cytokinesis. Now, big differences between mitosis and meiosis is that there is only chromosome replication happening in the beginning for mitosis, where every time it goes through mitosis, it will go through chromosomal replication. But when it happens with meiosis, what happens is that you have chromosomal replication only occurring before prophase 1, not prophase 2 or meiosis 2 or anything like that. And with mitosis, the daughter cells are genetically identical to each of the each other and to its parent cell. Whereas in meiosis, what happens is that you end up with 4 haploid cells that are genetically unique from each other and the parent cells. So now what happens is that these haploid gametes come together and become like eggs and a sperm. And what happens is that they fuse together in a process called fertilization. And what happens is it basically complements the entirety of the chromosomes themselves, which basically brings it back to being a diploid. In this case, the cell that results from this whole process is called a zygote, and it's diploid. A zygote has a maternal and a paternal set of chromosomes. So that's what ends up becoming later on as like a little individual or a baby or an offspring, so on. So now what we have is um, this process here where we have gametogenesis. So from it being the gametes, what happens is that they go through a process called gametogenesis, which it slowly goes through the process of forming sperm or eggs, which is basically called spermatogenesis for sperm production and oogenesis for creating the ovum, which is also referred to as the eggs. So in spermatogenesis, you end up with at least four. In oogenesis, you get one egg out of every um, uh, oocyte that's produced. And you get three polar bodies in the process. So now, how does this end up creating a greater amount of variety? Like meiosis itself has been shown through many different processes that it can be leading to so much variability within the same species. And that's because when we started off, there's the process of recombination that basically shuffles up the alleles so that some from each parent gets passed on. And then there's the two rules between Mendel's ideas where independent assortment occurs during meiosis 1, whether which chromosome ends up with what. Basically, if it would be in an egg or a sperm. And then the segregation of alleles occur during meiosis 1, where basically one of each chromosome pair is passed on. So it brings us back to Mendel's ideas in how genes and alleles get separated, and then the random pairing of these gametes during fertilization. It's entirely random in which egg or sperm will fuse together to end up with that new zygote of all the gamete cells that are produced within these organisms. In this case, one fact about independent assortment, as we know, it increases the amount of genetic variation. So in this idea where it's like if you pick any one of them, one of the pairs in each, and then into the sperm and one of each into the egg and form that zygote, in all of these you can have about 2 to the 23rd. So 2 to the number of chromosomes that an organism has is the amount of combinations you can have. So in this case, if you have 2 to the 23rd, because we have 23 sets of chromosomes, we get about 8.4 million combinations of chromosomes that you can have in the gametes themselves. And two parents can basically produce basically 70.6 trillion, because when you look at it, it's like 8.4 times 8.4, basically giving you that amount of genetically distinct offspring. You wouldn't think it's this high, but it's definitely is. So basically, if your parents end up having another child, or if you end up wanting to have another child with the exact same genetic makeup, it's about 70 trillion to 1. And it makes this is basically more than the number of humans who have ever lived. There is no one else in the planet Earth who has your same genetic makeup unless you have an identical twin. And in this case, sexual reproduction overall just leads to greater variation compared to like a asexual reproduction like mitosis. They are like clones. But then with sexual reproduction like these, it's like they're genetically different from each other and their parents, like these cute little doggies over here. So now when things go awry, there will be some aspects where you have to consider about chromosomal abnormalities. One of the first ones that would be included is aneuploidy, which is basically either too many or too few chromosomes in a cell. And it can result from the wrong amounts of protein being produced in this scenario. Uh, Basically, if you have any chromosomal abnormalities, you end up with severe um, issues in like having like health and like basically have non-viable gametes or embryos. And essentially mistakes in meiosis can end up resulting in gametes with abnormal chromosome numbers. They can be structural, they can be number wise, depending on what step it ends up being affected. So if it's like n+1, it's like too many, or like if it's like n-1, it's like too little. So it just kind of occurs through non-disjunction, which is basically the uneven pairing of chromosomes. So it ends up with many different issues, basically resulting in those kind of conditions. So now aneuploidy has a couple of different terms. In this case, there's monosomy, which is one less than the normal set, which basically means that they are missing one chromosome. It's 45 chromosomes instead of 46, and it's just an it's an X chromosome that's usually missing. These result in Turner syndrome, which is basically a female that's sterile. And then there's trisomy, which is an extra copy of a chromosome. So like 2n+1. So like examples would be like Down syndrome, where it's like 47 chromosomes, where it's an extra chromosome of chromosome 21. Or trisomy 13, where it's an extra chromosome of chromosome 13. Or Klinefelter, where it's an extra X chromosome. And then there's a whole nother set of uh, chromosomal abnormalities where it has an entire set added, where that's referred to as polyploidy. So that's basically an entire set of chromosomes getting added on. So like if you have 3 times the number of chromosomes or 4 times the number of chromosomes. So like 3N, 4N, 5N, which is like triploidy, tetraploidy, and pentaploidy, and so on. So now polyploidy, what happens is that, like, as we were talking about, it's typically more than 2 sets of chromosomes where the chromosomes just fail to separate during non-disjunction. It's not really the best thing in humans, but it can play important roles in like plant diversification, speciations, and kind of just get extra copies of the genes that can yield new traits and species, but the odd number kind of leads to sterility. So like seedless varieties of certain plants, it's rare in animals, but it results in non-viable gametes or offspring that do not survive. Some animals show no ill effects, but primarily in humans, it's usually non-viable. Now, the frequency of non-disjunction occurring to actually have aneuploid zygotes is around 10% of meiotic divisions. It's usually they do not typically survive to produce a viable offspring, but it depends on like the case itself, like with Down syndrome and Turner syndrome and so on. So like with this, it's like these errors are basically the leading cause of spontaneous abortions or like miscarriages. Sometimes it's accidental. There's no um, genetic predisposition. And there have been times where the increasing maternal age kind of is linked to increased frequency of non-disjunction, and that's kind of there in terms of like the frequency of it occurring. So now trisomy 21, basically, as we talked about, it's an extra chromosome in chromosome 23. It's basically 90 greater than 90% chance that it occurs because of non-disjunction through oogenesis. Majority of the times it occurs during meiosis 1. The recombination errors end up leading entangled homologous chromosomes that end up not separating. And the main risk factor is the age of the mother itself at the birth of the child. And so there are a couple of other structural abnormalities now that we've kind of branched off from the chromosomal number abnormalities. These are basically in ideas of like deletion, duplication, inversion, substitution, translocation, and sometimes ring chromosomes. So it's like going from like deletions where a part of it gets deleted, parts of it where it's getting duplicated, flipped around, substituted from another chromosome, translocated from another chromosome into another chromosome, so on. It's like these different abnormalities can occur, and it usually ends up with various scenarios of like diseases and stuff like that. So now we go into the idea of XN inactivation. XN activation is basically having the cells. So in this case, having cells with. So one thing about cells in general, if you don't have the correct number of chromosomes, like we saw, it it usually ends up with like different diseases and like outcomes that you do not want. So why is it that females, when females themselves have two X chromosomes and males only have one, why isn't that a problem? And that's because there's a process itself called X inactivation, where humans and many other animals only need one X chromosome for the information. So what happens is that the second X chromosome gets turned off permanently in every cell very, very, very early on in development. And the random exon activation itself, it's basically the maternal or paternal one is randomly turned off in different cells. And descendants of those cells continue to express only the genes from that X chromosome that's still active. So like these calico cats or like those where it's like black and orange fur. One of the X chromosomes is inactivated, and then it ends up like this mosaic pattern where we end up with the cute little tortoiseshell cat. It's also seen in calico cats where they are primarily female because you need both X chromosomes. If these cats basically inherit both orange and black coat fur, they're like this guy here, then different parts of the fur will express the fur color allele that is still active. So in areas that it just goes through there. But if there are males that male calico cats, it's because they have an extra X chromosome, because that's the only way that this pattern would show up. Now in here, it's like if it ends up with different alleles on each X chromosome, they can express that mosaic pattern like our calico cats. And what happens is it's like with the lack of sweat glands in heterozygous females, what happens is the pattern shows up wherever the X chromosome is inactivated. So that's how mosaicism kind of shows up in like cats, humans, so on. And with that, we conclude this topic for this week, and we will pick it up next week more on the central dogma of biology.